Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for October 2022. Let us begin with the first question. Question 1. The graph shows a bacterial growth curve produced by counting the number of bacterial colonies using the dilution plating technique. So this is the graph given to us. They say which label should be used for the y-axis of this graph. The y-axis is logged to base 10, the number of bacteria, so the answer should be A. Moving on. In part B, when culturing microorganisms, it is important that an aseptic technique is used. Explain the importance of using an aseptic technique. Aseptic techniques are used to eliminate any unwanted microorganisms because if these unwanted microorganisms are present, they will compete with the microorganisms of interest for nutrients as well as space. Also, these aseptic techniques ensure that pathogenic microorganisms are not cultured because these can cause harm or disease. The next part says, explain two aseptic techniques that should be used in dilution plating. We could use autoclaving, whereby all the apparatus as well as the agar are sterilized before use. Also, flaming of the inoculating loop and the necks of bottles with a Bunsen burner flame to prevent contamination. You can also wash hands and use sterile gloves. And finally, reducing exposure of the culture to air, since air contains some unknown microorganisms. So this brings us to the end of question 1. Let's continue to question 2. Question 2. The following features of viruses can be used to classify them: the type of nucleic acid they contain, the arrangement of the proteins in their capsid, the presence or absence of an envelope, the enzymes they contain. Part A says, for each structure, put one cross in the appropriate box in each row to show which viruses have that structure. For DNA is contained by neither Ebola nor HIV. The helicocapsid is by Ebola virus only, and the envelope is for both Ebola and HIV. In part B, human cells contain several types of DNA polymerase. One type of DNA polymerase, found in some cancer cells, has been shown to synthesize DNA from an RNA template. Name the enzyme found in some types of virus that can synthesize DNA from an RNA template. This is called reverse transcriptase. The next part says, suggest why this DNA polymerase may be a target for drugs used to treat cancer. This is because only the cancer cells containing that DNA polymerase will be killed by the medication, so normal cells will not be harmed by that treatment. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Chloroplasts are involved in both the light-dependent reactions and the light-independent reactions of photosynthesis. Which row of the table is correct for these two reactions? The light-dependent reactions, ATP is produced, so ADP is phosphorylated, requiring energy. In the light-independent reactions, ATP is hydrolyzed or broken down, and energy is going to be released. So here B is going to be our answer. The next part says, the diagram shows part of a chloroplast. So if I take away what I answered, you will be able to visualize what was present there initially. So this is how the chloroplast was. They had labeled for us the stack of thylakoids as well as the outer membrane. And they said, complete this diagram to show 3 other labeled structures found in a chloroplast. So this is how I labeled everything. As you can see here, I input the DNA, the ribosomes, inner membrane, the stroma, starch grain, the melee or what you call the integral membrane. So if you had put any 3 of these, you would get full marks. Moving on. Here they say, compare and contrast the structure of the outer membrane of a chloroplast with that of a thylakoid membrane. So since both are membranes, I began with similarities. Both are made up of phospholipids, and both contain molecules in their membrane like channel proteins and functional proteins. The differences are that the thylakoid membranes are greatly folded, while the outer chloroplast membrane is not. The thylakoids contain chlorophyll and photosystems, while the outer chloroplast membrane does not. The thylakoid membranes contain the electron transport chain and ATP synthase, while the outer chloroplast membrane does not. So this brings us to the end of question 3. Let's continue to question 4. Question 4 Scientists have used specialized apparatus to remove cores of mud from a tropical rainforest in the Republic of Congo. The diagram shows the apparatus containing a core of mud. This is the mud that has been sampled. Down here they say, the core of mud removed has a diameter of 80 millimeters and a length of 900 millimeters, which is the volume of this core of mud. They say use the formula volume is equal to pi r squared L, where pi is 3.142. Since all answers are in centimeters cubed, I had to convert this into centimeters. 1 centimeter is equal to 10 millimeters, so 80 millimeters is 8 centimeters and 900 millimeters is 90 centimeters. So volume is pi r squared L, which is the pi given to us here, times the radius squared, which is going to be diameter divided by 2 and then square that, and then times the length. So using the calculator, the answer came out as 4524 centimeters cubed, and A is our answer. Moving on. In part B, the scale diagram shows where 3 types of pollen grains were found in a core of mud. The depth of mud is proportional to how long ago the mud was deposited. So here they've given us this is the present day, and this is 2,700 years ago. So down here they say, calculate how many years the plants producing type D pollen grains were present in this rainforest. So we can see this type was present for this period of time. All I did was to find the length, and it's about 2.5 centimeters. I also found this length because this is the present day and this is 2,700 years ago. So this distance can give me the scale that corresponds to 2,700 years. So when I measured, the length was 9 centimeters. So we can confirm that 2,700 years ago, or you could say 2,700 years, is equal to 9 centimeters. So 1 centimeter is going to equal to 300 years, or you could say 300 years ago. So type D is present for 2.5, so that means if 1 centimeter is equal to 300 years, then 2.5 is going to be 2.5 times 300, which is 750 years. So this was my answer. This part here corresponds to 750 years. Moving on. Here they say, explain the distribution of these 3 types of pollen grains in this mud column. Use the information in this diagram to support your answer. If I take you back here, you can see different types of plants were present in different time periods, and that means different conditions favored the growth of specific types of plants. So here I said, the type of pollen deposited shows the type of plants that were favored to grow under specific conditions. Type F was present before other types and it was deposited for more years, meaning you can see the time period is longer in comparison to these two. This shows that plants with pollen F survived long, and during the time, the conditions did not change drastically. However, changes in conditions allow plants with pollen D and E to appear. This led to the disappearance of species F. It was outcompeted, or the conditions were no longer favorable for its growth. Due to change in conditions, disease or competition for space, it could be competition for nutrients, sunlight and water, both species D and E disappeared as well. So this brings us to the end of question 4. Let's continue to question 5. Question 5. Human serum albumin is a globular protein found in blood plasma. Albumin has a molecular mass of 66,300 daltons. It consists of 585 amino acids. At pH 7.4, albumin has over 200 negative charges on the surface of each molecule. Remember, the negative charges are due to carboxylate groups. So they say, calculate the mean molecular mass of an amino acid in albumin and give your answer to an appropriate number of significant figures. So the mean molecular mass is equal to the molecular mass of albumin divided by the total number of amino acids. So we were given this 66,300, and this is the number of amino acids. So 66,300 divided by 585, and this is going to be our answer here. Next they say, explain why albumin is soluble in blood plasma. So I said, albumin is negatively charged, so it will form ion dipole interactions with water molecules in plasma and dissolve. Albumin is polar, so it forms intermolecular hydrogen bonds with water molecules. In part B, albumin can be separated from other proteins in blood plasma A1, A2, B, and G by gel electrophoresis. The diagram shows the binding protein produced on separation of these proteins by gel electrophoresis. So this is the origin with CG, B, A2, A1, and albumin. When we use gel electrophoresis, we will observe the size of the band as well as the position of the band along the gel. So here down they say, protein G has a concentration in blood plasma in the range of 0.700 gram per decimeter cubed to 1.700 gram per decimeter cubed. Albumin has a concentration in blood plasma in the range of 0.525 gram per decimeter cubed to 1.275 gram per decimeter cubed. They want you to calculate the maximum difference in the concentration of these two proteins. So maximum difference is going to equal to the maximum concentration of G minus the minimum concentration of albumin, which is going to equal to the maximum here, which is 1700, minus the minimum here, which is going to be 0.525, giving us 1.175 gram per decimeter cubed, and therefore this is my answer here. Moving on. In part D, describe the conclusions that can be made about albumin compared with the other 4 proteins present in blood plasma. Use the information in the diagram to support your answer. So taking you back here, since albumin traveled farthest from the origin, it means it had the greatest solubility, and G had the least solubility because it traveled the shortest distance. So I said, albumin moved the farthest, so it has the greatest solubility in the solvent used. Albumin was farthest from the origin, so it could be the lightest, meaning the least molar mass. Also, albumin has the thickest band, so it possibly has the highest concentration. Moving on. In part C, albumin binds to and activates a protein channel HHV1. This protein channel is present in the membranes of sperm cells. When the channel is activated, sperm cells have the ability to penetrate and fertilize egg cells. Sperm cells are released in a liquid called seminal fluid. Seminal fluid has an albumin concentration of 15 micromole per decimeter cubed. The fluid inside the uterus of the female has an albumin concentration of 500 micromole per decimeter cubed. Explain the role of albumin concentration in controlling fertilization. The seminal fluid contains a lower concentration of albumin in comparison to the female, and that is to ensure that sperm cells are not activated before they enter the female. So here I said, since fertilization takes place in the female, who has a concentration of about 500 micromole per decimeter cubed albumin, the 15 micromole per decimeter cubed found in the male is not sufficient to activate the sperm, so sperm is not activated until in the uterus of a female. This avoids wastage of sperm and energy for activation before the fertilization time. It also ensures that only the sperms that are viable enough to swim to the uterus are activated. This increases the chances of fertilization being successful. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Alzheimer's disease is a cause of dementia in older people. The neurons in the brain are damaged and eventually destroyed. Abnormal plaques are seen in the brains of people with Alzheimer's disease. These plaques are made of a protein called beta amyloid. Drug ATD is a new drug used to treat people with Alzheimer's disease. This drug is an antibody that is specific to beta amyloid. Part A says, drug ATD was recently approved for the treatment of Alzheimer's disease. Describe the information that had to be collected by scientists before this drug could be approved. Since the intention of this drug is to decrease the size of the plaques, they have to make sure that the drug works by decreasing the size of the plaques. They have to make sure they find out if there are any side effects in the body of the humans. They have to determine the appropriate dosage or concentration that has to be given to the people. And they have to make sure they find out how long the treatment has to last. So I said information on whether the drug causes side effects on humans, information on whether the drug reduces the size of the plaques, information on the right dosage or concentration of the drug to be given, information on how long the treatment can last for all beta amyloid to be destroyed, and information on how the drug can be administered. Is it going to be orally or intravenously? They have to find out about that as well. In part B, explain the phrase, an antibody that is specific for beta amyloid. When they say antibody that is specific to beta amyloid, it means it's going to bind onto the surface of beta amyloid. So I say the antibody can only bind to specific antigens or surface molecules on beta amyloid, and this is because the shape of the antibody is complementary to the shape of the antigen found on the beta amyloid. Moving on. So here they say, drug ATD reduces the buildup of beta amyloid in the brains of people with Alzheimer's disease. Suggest how drug ATD could result in the reduction of the buildup of beta amyloid. Antibodies can cause agglutination. So these antibodies on the ADT drug will bind onto antigens of the beta amyloid, and this is going to cause macrophages to engulf the amyloid proteins. Due to the agglutination, macrophages are going to engulf them in large numbers. Then the amyloid proteins are then destroyed through lysosome action as the macrophages release lytic enzymes. In part C, drug ATD was derived from a B memory cell taken from an elderly person who showed no signs of Alzheimer's disease. Suggest why this person did not have Alzheimer's disease. This is because this person's immune system was already triggered against beta amyloid, so the amyloid had already been broken down through phagocytosis by macrophages. Next they say, explain why this drug could be produced only from B memory cells taken from a person who did not have Alzheimer's disease. Because if a person had Alzheimer's disease, then they will not have the B memory cells, so no differentiation to produce B plasma cells that have the potential to produce antibodies. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Methicillin-resistant Staphylococcus aureus is a disease-causing worldwide concern. Methicillin is one of the antibiotics to which this pathogen have developed resistance. Castaneroxy A is a molecule extracted from the European chestnut tree endemic to southern Europe and Turkey. The effects of Castaneroxy A have been investigated. The investigations have found that Castaneroxy A does not affect the natural skin flora. It prevents MRSA from producing toxins. It weakens the MRSA bacteria and reduces the size of skin infections caused by MRSA. So the first question says, explain why the development of this drug is an example of an evolutionary race between humans and this pathogen. This is because MRSA have already developed resistance against methicillin, so a drug that bacteria are not resistant to has to be found. So this is an evolutionary race. The next part says, explain why it is important that Castaneroxy A does not affect skin flora. Skin flora is advantageous in outcompeting the pathogens on the surface of the skin for space and nutrients. So if they are outcompeted, then it means we're going to be free from infection. So I say it because skin flora helps to prevent growth of pathogenic bacteria by competing with them for space and nutrients. In part C, suggest how weakening the MRSA bacteria could help the recovery of a patient infected with this bacteria. If bacteria are weakened, then the growth rate is going to be slower, meaning this gives time for the immune system to be able to fight. So I said if the bacteria are weakened, their growth rate or rate of reproduction will be slower or lower, so fewer toxins will be produced and the immune system will not be overwhelmed, resulting in effective response. In part D, in an investigation, patients who had developed skin infections were divided into 3 groups. 2 groups were each treated with a different concentration of Castaneroxy A. The other group was treated with only the solvent that was used in the treatment. The graph shows the results of these treatments on the mean size of the skin infections. So on the vertical axis, we have the mean size of skin infections, and on the horizontal axis, we have time after infection of skin with MRSA, and the time is in days. So we can see with solvent only, the mean size of skin infections increase, and then they'll decrease, but the maximum is reached on day 6, and it has a very high mean size. When we see these 2 concentrations of Castaneroxy A, the higher concentration has a lower mean size of skin infections in comparison to the one that is 10 microgram per centimeter cubed, meaning the higher concentration of Castaneroxy A is more effective than the lower concentration. So here they say, explain why this investigation included treatment with the solvent only. The one for the solvent only is for comparison so that we can know that the difference or observed effects are going to be solely due to the drug that has been used. So I say to measure the size of the skin infection with the solvent only, meaning for comparison, so that the effect only due to the drug can be measured. Next I say, determine the effect that castaneroxy A has on the mean size of skin infections. I've already talked about the results from the graph. So the higher the concentration of castaneroxy A, the lower the mean size of skin infections. Here we can see with a higher concentration, which is this, we have lower mean size in comparison to this that has a lower concentration. And then castaneroxy A does not prevent increase in size of the skin infections, but its increase is not as high as the increase with the solvent. This means, based on the results we see here, even with a high concentration of 50 microgram per centimeter cubed, we still see some skin infection appearing. So a higher concentration causes a decrease in the mean size, but it does not fully prevent skin infections. So the increase is not as high as the increase with a solvent only. Also by day 6, the difference in the mean size of skin infections at both concentrations of castaneroxy A is about 0.5 to 5 per centimeter squared. If you can look at the difference, we have 0.375 and 0.05. The difference in area is going to be 0.525 per centimeter squared. So here they say the leaves of the chestnut tree contain 0.0019% by mass of castaneroxy A. Calculate the mass of leaves in kilogram that would be needed to produce 1 decimeter cubed of castaneroxy A at a concentration of 50 microgram per centimeter cubed. They want you to give your answer to the nearest kilogram. So for concentration of 50 microgram per centimeter cubed, we can say 1 centimeter cubed contains 50 microgram of castaneroxy A. You know 1 decimeter cubed is 1,000 centimeters cubed, so this is going to contain 50 times 1,000 microgram of castaneroxy A, which is equal to 50,000 microgram of castaneroxy A. Now based on the percentage composition, 0.0019 microgram of castaneroxy A is going to be contained in 100 microgram of leaves because they said 0.0019% by mass, meaning if the mass is going to be 100 microgram of leaves, the mass of castaneroxy A is going to be 0.0019 micrograms. So this is the concentration I'm using here. So 0.0019 microgram of castaneroxy A is contained in 100 microgram. That is using that. That means the 50,000 micrograms of castaneroxy A are going to be contained in 100 divided by that times that, giving me this, and that is my answer here. But remember, this is in microgram, but we need the answer to kilograms. So 1 microgram is equal to 1 times 10 power negative 9 kilograms, and multiplying this answer here times 10 power negative 9, I got 2.631578947 kilograms. To the nearest kilogram, it's going to be 3 kilograms, and that is my answer here. So this brings us to the end of question 7. Let's continue to question 8. Question 8. There has been an increase in the number of forest fires in many areas of the world. Climate change caused by humans is claimed to be responsible for this. The photograph shows the devastation caused by a forest fire in Myanmar. So state the term used to describe climate change caused by human activity. This is called anthropogenic climate change. Then explain why some people consider this claim to be controversial. This is because the evidence that is available only shows a correlation but not a causation. There is no direct link that climate change is caused by human activity. It's only a correlation. Also because some forest fires are not caused by human activity. And lastly, because some people, like politicians, have different opinions on the matter, so they could say certain statements in order to satisfy their political goals. Moving on. In part B, discuss the effects that forest fires have on the local atmosphere and local biodiversity in the short term and in the long term. So we have short term and long term. Forest fires are going to increase the concentration of carbon dioxide in the atmosphere, so global warming could result. There is going to be an increase in the destruction of organic matter, and a lot of airborne particulate matter, which is soot, is going to be produced. There will be a decrease in the number of trees, which will lead to less photosynthesis. The biodiversity will initially decrease. This means in the short run, there will be less food, shelter for animals will be destroyed, some animals are going to be killed in the fires, and the food chain is going to be affected or completely destroyed. Also, the gene pool will be affected since fewer organisms may survive to reproduce. This means a lot of inbreeding is going to occur. Also, habitat fragmentation will take place. That is for the short term. However, over time, seeds will begin to germinate, new species may arise as secondary succession takes place, and more photosynthesis will lead to a low atmospheric carbon dioxide concentration, more food and shelter will be available, and biodiversity will increase because more organisms are going to come to the newly formed habitat in order to get shelter as well as food. In part C, reforestation is one way to repair the damage caused to forests by climate change. Explain why the changes to the environment caused by climate change need to be considered when selecting the species of trees to use in reforestation. Remember, when destruction of trees has occurred, the habitat is going to be changed. There will be no more trees to compete with for sunlight. So when we are planting the new trees, we have to make sure we consider so many biotic and abiotic factors that will lead to the faster regeneration of the habitat. So I say the new conditions may not be suitable for the original trees, so new species of trees can be planted. For example, the pH of the soil could have changed, the water content in the soil could have changed, so planting the same trees that existed initially may result in failure for those same plants to grow in that area, so new species may need to be planted. Also, the trees should be well adapted for the new conditions. The new trees should prevent loss of minerals through leaching by holding the soil together, meaning the new trees should have a good root network system. Also, if trees with higher productivity are planted, there is a high chance of faster restoration of the habitat and more CO2 removal from the atmosphere. So this brings us to the end of question 8. Let's continue to question 9. Question 9. Plants store biomass both above and below ground. Scientists have found that on average, 24% of plant biomass is underground in the roots. The total plant biomass underground contains 113 gigatons of carbon. This is equivalent to the mass of carbon dioxide emission produced by humans in 10 years. 1 gigaton is 1 times 10 power 12 kilograms. They want you to calculate the mass of carbon dioxide in kilograms produced by humans in 1 year. And they want you to give your answer in standard form. So that means CO2 produced in 10 years is 113 gigatons. But 1 gigaton is that, so 113 times 10 power 12, this is going to be the mass in kilograms. So CO2 produced per year is going to be that divided by 10 because this is for 10 years, giving us 1.13 times 10 power 13 as the mass of CO2 produced per year in kilograms. In part B, describe how gall produced in the leaves becomes incorporated into biomass in the roots. If gall is produced in the leaves, it could be converted into glucose or fructose, and these are going to be used to produce sucrose in the leaves. The amino acids are also going to be produced. Then sucrose and the amino acids are going to be transported through the phloem to the roots, where they convert it into molecules like proteins and starch through condensation reactions, and then those molecules can be stored as biomass in the roots. Moving on. In part C, inorganic ions are used by plants to make molecules. The table shows some molecules made by plants. For each molecule, put one cross in the appropriate box in each row to show which inorganic ion provides an atom found in the molecule. This is only carbon, hydrogen, and oxygen, so it's going to be neither nitrate nor phosphate. Nucleic acids have carbon, hydrogen, oxygen, nitrogen, phosphorus, so we can say both nitrate and phosphate. And triglycerides have only carbon, hydrogen, and oxygen, so we can say neither nitrate nor phosphate. Moving on. In part D, forests, shrublands, and grasslands are 3 types of ecosystems. Biotic and abiotic factors affect how much biomass is stored in the roots of plants. The graphs show the importance of some abiotic factors in determining how much biomass is stored in the roots of these ecosystems. So with the forest ecosystem, we can see the yearly mean temperature is the greatest determinant, then soil moisture content, and then the lowest determinant is going to be depth of the soil. Then when we go to shrublands, we can see also yearly mean temperature is a great determinant, and the least determinant is going to be depth of the soil. Then for grasslands, we see the soil moisture content is going to be the greatest determinant, but the least is going to be depth of the soil. This is similar for all the three types of ecosystems that we have seen. So here they say, discuss how these factors could affect the proportion of biomass stored in the roots of plants growing in these three ecosystems. So in my answer, I wrote the specific conditions like 1, 2, 3, 4, 5 for all the ecosystems together. So I say, yearly mean temperature affects biomass storage in all ecosystems, but mostly in forests and shrublands. This is again seen here. Forest is the highest, shrubland is the highest, but when we go to grasslands, this is the highest. Moisture content is more preferred. Then I said, photosynthesis is enzyme controlled, and a higher temperature leads to a higher rate of photosynthesis and a faster production of GALP in the light-independent reactions. When we go to soil moisture content, this has the greatest effect on grasslands, followed by forests. Again, to take you back here, we can see it's going to be the highest in grasslands, and then we can see followed by that, and then this is going to be the least. This is because water is required in the light-dependent photolysis reactions. Water is also a transport medium for mineral ions and sucrose, or you could say amino acids. When we go to density of trees, this affects all ecosystems, but mainly forests. A high density leads to competition for nutrients, light, and minerals. Competition for sunlight can lead to slower light-dependent reactions, less ATP, less reduced NADP, so less GALP production and less biomass storage can occur. Then the carbon-nitrogen ratio, this affects all three ecosystems. Plants need nitrogen for amino acid synthesis, protein as well as enzyme production. An example of an enzyme is rubisco, which is important in the fixation of CO2 that is helpful in the process of formation of GALP. The depth of the soil affects trees the most, and this is because soil with greater depths allow for stronger tree anchorage and extensive root networks. This requires more biomass storage in the roots. The last statement means the depth of the soil is important because for trees to be anchored properly in the soil, a good root network system is required. That is going to be beneficial for the forest ecosystem. So this brings us to the end of question 9 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.